Yay, you made it to the end. It's our third and final video, and we're going to go over the stages of mitosis with a much more a closer eye on the nitty-gritty details of what's happening. So in that last video, we were looking at the stages of mitosis and just sort of briefly introducing the names of our four phases, which are prophase, metaphase, anaphase, and telophase. And in this video, we'll go into more detail about what exactly is happening at each of these stages. So I'm just going to back up slightly to make sure we're all on the same page about where we're starting. And remember that before mitosis even happens, or before it even starts, the cell is going through different parts of interphase. So we had the G1 phase where the cell is growing and doing cell stuff. And then it goes to the S phase where the DNA is replicated. And then we end up in G2 where the cell is growing some more. And after G2 is where we're kicking off mitosis from. So remember that in G2, all of our DNA is still in its chromatin form. So it's still unwound, it's very loose and unpacked. And since we've already passed through the S phase, all of our DNA is replicated. So all of our chromosomes are copied. So if we're thinking about taking inventory of our DNA when we're starting mitosis, we would say that it's in chromatin form rather than chromosome form, and we would say it's replicated rather than unreplicated. Okay, so you'll notice that the DNA is contained in the nucleus to sort of keep it safe, and that's where we're going to start with. So in our first stage, we said it was called prophase, and here is prophase. This was all G2 of interphase. So we've got our chromatin and it's all replicated. So starting with prophase, there are a couple things that are going to start happening. So this is basically a phase where we're sort of prepping to really get into mitosis. It's the beginning of mitosis, so there's a lot of preparation involved. So one thing that you'll maybe notice is that the nuclear membrane or the nuclear envelope is starting to break down. So these two pictures are showing the beginning of that, and this is just sort of the end of prophase. So we started off with a nuclear envelope, and then we're ending with just sort of these fragments. Um, and this makes sense if you think about the fact that our chromosomes are going to have to be divided up between two daughter cells, and they're going to be pulled in opposite directions of the cell. And we can't do that if our chromosomes are trapped inside of the nucleus. So if we're trying to get them to move around and get them to the places that they need to go, we need them to be able to move freely. So disintegrating the nuclear envelope or the nuclear membrane lets us do that. Another thing that's happening is our chromatin is condensing and turning into a chromosome form. So you can see it sort of in between here. At the beginning of we have condensation, and then by the end of prophase, we are fully condensed into a chromosome form. And again, this is just to make it easier for the cell to keep track of where the DNA is going and make sure that all of the chromosomes get to the right place because they're in this compact, super coiled up form. And then last but not least, we also have the formation of what's called the mitotic spindle. So early mitotic spindle here, um, and then more fully developed spindle here. And these are sort of like tracks or arms that are going to move the chromosomes around. So the spindle is going to move the chromosomes to where they need to go. And in order to do that, it's actually going to connect to the chromosomes and physically move them around. So to recap, in prophase, we get disintegration of the nuclear envelope. We get condensation of our chromatin into chromosomes. And finally, we're making our mitotic spindle. And then next comes metaphase. And I think this is kind of the easiest one. It's definitely the easiest one to visualize and identify because basically all that's happening in this phase is that all the chromosomes are lining up in a single file line right across the middle of the cell. And people call this either the metaphase plate or the equator, but really all that you need to know is that they're lined up in a single file line And this just sort of lets the cell keep track and make sure that it has all the chromosomes and that everything is in order for mitosis to move forward at this point. Also, you'll notice that all of the spindle fibers are going to be connected to the chromosomes, and that connection happens at the centromere. And maybe you remember that the centromere is what's holding these two sister chromatids together. So, if we're looking at this one chromosome, this one replicated chromosome, we've got a spindle fiber attaching to this sister chromatid on the top side, and we've got another piece of the spindle connecting to this sister chromatid on the bottom side. So we've got spindle fibers on both sides attached to both sister chromatids at the centromere of the replicated chromosome. Okay, so that's metaphase, is that lining up. And then our next phase is called anaphase. And this is where the magic is starting to happen. So at this point, we're going to pull apart our sister chromatids. So they're not going to be attached to one another anymore. So this used to be the sister chromatid on top, and this used to be the sister chromatid on the bottom. So they used to be sister chromatids, but now that they're on their own, now that they're individual, we're going to call them chromosomes now. So no longer sister chromatids. Now we have individual chromosomes that are going to go live in separate daughter cells. So the sister chromatids are pulled apart. And again, that's happening because these mitotic spindles are attached to the centromere. So we get the pulling apart of sister chromatids in anaphase. And then finally, our last phase of mitosis is called telophase. And then right after this, we'll talk about a process called cytokinesis in the next slide. But telophase is happening when these chromosomes that we just pulled apart are going to take up new residence in the daughter cell that they're going to live in. So they're going to start to unwind again, and they're going to start to go back into that chromatin form. So it's sort of like we're going back to the beginning of where we started in G1. And then also, cells in general need nuclei regularly. Like we started with a nuclei, so we're going to rebuild that nuclear membrane or that nuclear envelope around the DNA. And then finally, for our spindles, we don't really need the spindles anymore because the DNA all got to its proper place. So the spindle fibers are going to go away and they're going to just kind of disintegrate. So this is going to happen on both sides, and we're going to get a new nucleus over here and over here with the DNA. And that's what telophase is this portioning out of the genetic material and reforming the nucleus. So simultaneously with telophase, the actual cell itself is going to start to divide. And so the cytoplasm, or the interior contents of the cell, is going to start to split in half, and the cell membrane is going to start to pinch inward. And this is called cytokinesis. And it's not technically a part of mitosis because it doesn't have to do with the genetic material, but it usually happens concurrently or at the same time as telophase. So I just want to reemphasize that telophase has to do with the nucleus and the DNA, and cytokinesis has to do with division of the other cellular contents. So let's look at that process of cytokinesis for the last slide in this third video. So like I said, cytokinesis is going to refer to the division of the cytoplasm and the organelles between the daughter cells. And so it's not technically part of mitosis, but it usually does overlap with telophase. And there's a little bit of a difference between how plant cells and animal cells will do this because plant cells have a cell wall while animal cells don't. So they just go about it in slightly different ways. But the end result for both of them, for cytokinesis, is to actually physically separate the two daughter cells. So here, when we're done with telophase, we're going to have one individual cell with two nuclei. So we've got DNA here and DNA here. But they're not two separate daughter cells yet because they're still connected together, like in one cell. So it's one cell with two nuclei, but cytokinesis will allow us to form this contractile ring between the two would-be daughter cells, and you can it sort of just like pinches them off from one another so that they're separate from each other. And you can see this in an actual cell here. And so you can see it's literally just like squeezing the two cells apart until they separate into individual cells. And then in plant cells, they can't really squeeze them apart in the same way because they have these sturdy cell walls that provide a lot of structural support. So the squeezing is really just not possible, like you can do in an animal cell. But instead, what the plant cells do is basically just make a new cell wall right in between of the two daughter cells, and they separate them in that way. So here's another picture, and you can see this new cell wall is starting to form um, in between the two nuclei. So it's developing, and we have these membrane sacs that contain cell wall material, and they're forming this. We call it a cell plate when it's sort of in the process of being formed, and then as it continues to develop and grow, eventually it's a full-fledged cell wall that's just completely separating our two daughter plant cells. So that's cytokinesis, and it starts during telophase, but it's not a part of mitosis. And it has to do with the division of the cytoplasm, whereas mitosis is division of the nuclear genetic material. Okay, you made it through all three videos, and I know this was a lot of lecturing, even though it was split up. So we'll be practicing with this more in class and getting to see if we can identify these different stages. So I will see you all soon.